Sieg, and I'm a visiting assistant professor of biology at Middle Tennessee State University. And today I'm going to be talking about ways that I'm trying to change up the way we teach introductory biology through authentic research experiences tied to the Small World Initiative. So the issue that we're dealing with here is that a lot of students that are coming into college thinking they want to major in science or mathematics end up actually dropping out, usually in the first year or two, because they lose interest in the subject and switch to another major. So actually only about 40% of students intending to major in STEM actually complete that STEM degree. And you can attribute this to a couple of different factors. Two of those are that introductory labs tend to be what we call cookie cutters, meaning that each lab is kind of its own little thing. It's more like following a recipe than actually doing authentic research. And so students start to lose interest. Additionally, several students are coming into the university system with insufficient laboratory experience or math experience. And so once they hit an actual college level science class, they feel really threatened or challenged because they're not really sure what's going on. And so this schematic sort of shows that issue in a really sort of pretty way. But what we're seeing is that on the left-hand side, you can see what students are majoring in college, but then where they actually end up after they leave college. And so you can see that a lot of students are starting out in the STEM fields, but then they end up going into fields that have no relation to STEM whatsoever. And so what we're trying to do is find ways to make it so that they stay within those STEM fields upon graduation. And so the project that I'm working on emerged directly due to a presidential order released in 2012 by President Obama called the Engage to Excel proposal. And the basic idea here was to have a large-scale effort in the United States to increase graduation in our STEM fields by about a third, focus specifically on the first 2 years of college because those are the years when students are going to be most likely to switch a major. And also this was a uniform call across the university landscape, meaning that there's an equal focus on community college, small liberal arts schools, big research institutions, and everything in between. And our big goal here is aiming for retention. So can we keep our students in the fields that they're starting out in? And so there are many different recommendations within the Engage to Excel proposal. These are the 5 major ones. And if you'll notice number 2, we want the program called to replace lab courses with research courses. So take away the cookie cutters and bring in authentic research that is more similar to what students would do with a career in the sciences. And so again, this is that same recommendation: advocate and provide support for replacing standard laboratory courses with discovery-based research courses. So get away from the kind of boring cookie cutters and then be a little bit more like Neil deGrasse Tyson, where you can't hear you because science is just so awesome. So when the Small World Initiative founders started trying to find a way to engage students through authentic research, they looked for something that students would be familiar with in their daily lives. And the topic that they settled on was this emerging threat of antibiotic-resistant bacteria and the lack of new antibiotics coming through the drug pipeline. And so the figure on the screen right now is sort of showing you how we're seeing increasing resistance to current generation antibiotics, such as vancomycin. Yet there's a precipitous decline in the number of new antibiotics actually coming out of the drug pipeline. So the bacteria are getting more resistant. We're not developing new things to combat them. And this is something that comes up in the news quite a bit. So antibiotic resistance warnings remain unheeded. And then this one was particularly pertinent for last year when I piloted this class. Antibiotic resistance could be far worse than Ebola. So all of my students were freaking out over Ebola. I said there's other things we have to worry about as well. And so the program focuses on looking for new antibiotics out of soil. And this is a logical place to start, one, because it's easily accessible. All you have to do is go out in your backyard. But about 3/4 of the antibiotics that are currently in clinical use are derived from bacteria in the soil. And just with Streptomyces, which is a prolific genus within the soil, there's about 100,000 different natural products being produced by that particular bacteria. Not all of these are antibiotics, but it's just showing that these bacteria are capable of making some really interesting chemical compounds. And despite the ubiquity of soil, not all bacteria are easily culturable out of that soil, and so there are many species that remain to be discovered. This came to light just this last year, where the first new antibiotic in the past several years to start undergoing clinical trials came out of a soil system. I think it was in their researcher's backyard. So when the researchers were trying to determine how to actually incorporate this antibiotic project and make it accessible to students, they decided to focus on looking for these antibiotics out of the soil. And this made a lot of sense because everybody has soil in their backyard, and about 3/4 of the current antibiotics in use are coming from soil bacteria. And so just with Streptomyces, which is one of the most common genera in the soil that can be isolated, there's about 100,000 different natural products. So that's not saying that every single compound that's produced is going to be an antibiotic, but it is showing how prolific some of these bacteria can be with regards to interesting chemical compounds. And despite our success finding antibiotics in the soil, there are a plethora of bacterial species that we're not able to culture yet. And this can be due to us not having the right kinds of media, the right kinds of conditions in order to make those bacteria thrive. And also, we're not really sure necessarily what triggers a bacteria to end up producing compounds that could be used as antibiotics. And so there's ongoing scientific research in trying to improve our ways in which we can culture these and cultivate these bacteria. And there's a recent success story just in the past year where a new antibiotic called Ticobacter was isolated from the researchers' soil in their backyard, I think, largely using new culturing techniques. So as we develop these new culturing techniques, we can start finding new bacteria. As we find these new bacteria, we can potentially find new antibiotics. And so there's sort of the background for you in terms of why this project was appealing. And so with this small world concept, the idea was our freshmen and sophomores, so those first 2 years of college, these students are going to lead the charge in our search for new antibiotics. And so they're going to be exposed to authentic research. They're going to be challenged. They're going to fail a little bit, but that's what real science is about. And our students are participants. They're not observers. They're not just looking at a beaker and saying, did it change color? Yes or no? Okay, check that box off. You're done with lab today. They're coming in. They're maintaining cultures. They're taking care of their bioassays. They might be failing, but then they're learning. That's part of the process of science. They figure out how to fix and remedy the problems and then go forward. And our whole class content is structured around this real world problem. It's something that the students can relate to more than putting a potato in a beaker of water. And so, as I said, the Small World Initiative was developed at Yale through the Yale Center for Scientific Teaching. And they've had 3 cohorts of training. And so what they did is they brought in cohorts of about 10 to 15 faculty from community college, small liberal arts schools, and R1 institutions. And they came to Yale for a week, and we were trained in terms of how to do the bioassays, how to survey students, use assessment instruments, things like that. And then we moved on from there. And so this project is the brainchild of Jo Handelsman, so I wanted to give her a quick plug. She's moved on to be one of President Obama's science advisors, so there she is in the corner shaking hands with President Obama. But this project has been in development for several years now, and we now have a network of about 60 different universities nationally and internationally using this model in some way, shape, or form. And so this map is a year out of date. I need to update it, but you can see just some of the different areas that we're actually doing the Small World Initiative project. We're everywhere from Hawaii to Vermont. We're also in Malaysia, Belize, England. We had an international training session this past year as well, so we're spreading out more and more. And so where do I come in with this? So I was trained in 2014, and I decided that I was going to use 2 sections of honors biology at Middle Tennessee State University to see whether or not freshmen coming in can use this model as a way to learn biology. And so I taught 2 sections. One was called Exploring Life. This was a class full of non-majors, lots of music industry, business, liberal arts type majors. And then I also taught a section of general biology. This is the first of 2 semesters of introductory biology at MTSU. I'd say between 50 and 75% of these students are planning on pursuing a degree in STEM, but not all of them are. But this is our majors sequence. And so you can see both of the classes, student populations down on the bottom. In our fancy new science classroom, you can see the nice monitors and workbench space. It worked out really well. And where we actually decided to get soil, it's largely dependent on what school, where the school is located and what the faculty want to do. And so I decided to focus in on a local ecosystem that we have in Middle Tennessee, which are cedar glades. So this is an image of a typical cedar glade. There hasn't been very much microbiological research done in these areas, but there are some interesting flora and fauna living within this environment. It's very short soil depth above a limestone bed. And so I thought this would be a great way for students to connect with their local environment and find a little bit more out about the microbiology within. And so in a nutshell, the small world design is pretty straightforward. The beginning of the semester, we went out to the Cedar Glades, students found some dirt. They brought that dirt back in and they learned the basics of how to use a balance, how to use a graduated cylinder as they characterize their soil, how to make dilutions, skills that biologists are going to need to learn early on. And then they took a sample of that soil and they cultivated a lawn of bacteria from it. So here's just a representative culture. And from that culture, students were able to work on diversity and dilution series, understanding the effects of dilution series, things like that. And students then were able to pick a suite of between 10 and 20 bacterial isolates from this plate, and they developed a master plate. And so this plate was used for the remainder of the semester, and these were the panel of bacteria that my students were going to survey for the rest of the semester. So I had a class of 20 students. They worked in pairs, so we had 10 of these master plates that were used for the remainder of the semester. So a large survey of bacteria. And so what they did then is we tested this panel of bacteria against similar genetic relatives to the nasty nosocomial or hospital infections. So I wasn't crazy enough or qualified enough to offer a bunch of introductory biology non-majors the chance to work with MRSA, but instead I can have them expose their panel of bacteria to bacteria that will respond in a similar way to MRSA in terms of their susceptibility to antibiotics. And so we had a panel of 6 safe alternative bacteria. And students were able to design different bioassays to figure out whether or not their cultured soil bacteria can inhibit these genetic relatives to the nasty bacteria. And so what you see on the corner there is an example of one of these bioassays. You can see a lawn, that sort of spread lawn is the tester strain we're testing against. We have a whole bunch of the soil bacteria spread out throughout the plate. And in the top left corner, you can notice that some of these bacterial cultures are inhibiting the growth of our tester strain, suggesting these might be good leads for antibiotic production. As I said, there are many different ways that students can screen their panel against the testers. The example I showed you is similar to what we see up in the top left, where we have a bunch of isolates and then a tester strain spread throughout. You can also do solid-solid bioassays. You can do liquid assays as well. But students have some creativity with regards to how they actually go about doing this. And then you can see on the bottom of the screen, I wasn't exposing students to these different bacteria, but I was exposing them to safe genetic relatives to these guys. And here's just another close-up of one of those bioassays. You can see at the top of the screen the kind of zone of inhibition we're looking for as a putative antibiotic-producing hit. So students were able to screen these guys for whether or not they're producing antibiotics, but they also got to do other skills that biologists are going to need. So when we started talking about biotechnology and molecular genetics, students did PCR analysis. They amplified 16S rRNA, and then they were able to actually do sequencing to identify what one of their most interesting bacteria was, at least to the genus. My background is in chemical ecology and natural products chemistry, so if we had a bacteria that had a strong hit for antibiotic production, we wanted to go from it's a culture inhibiting bacteria to maybe there's some chemistry behind this. So you can grow up a batch of the bacteria, take out the chemistry from that culture, and then separate it out, fractionate it out based on polarity or size, and actually try and isolate these compounds that are responsible for the activity that we're seeing. And then finally, we can also screen for antibiotic resistance. So maybe these bacteria in the soil are producing antibiotics, but they themselves could also be antibiotic resistant. So that leads to conversations about evolution and why antibiotic resistance could build up in the environment, things like that. And so here are just some sample results. So between my 2 classes, we isolated about 400 different colonies. Most of them were bacteria. I will be honest in saying that a couple of students ended up culturing fungi. So we started with about 400 bacterial isolates. 100 of these were screened against our suite of screened against our safe tester strains. And then from those 100 isolates, they were also all tested for resistance to common antibiotics. So we have kanamycin, gramicidin, and tetracycline. So antibiotics that target gram-negatives, gram-positives, or broad-spectrum antibiotics. This allowed students to try and characterize the types of bacteria they were culturing just a little bit more. And we had the students try and identify 20 of these 100 that we had separated. 90% of the students successfully amplified the 16S rRNA sequence by PCR. And then of that 90%, 80% successfully were identified at least to genus using sequencing. And so the kinds of things we got, there were a couple strains of Bacillus, Panibacillus, Variovorax, Ralstonia, a couple of other things too. Nothing novel and new, mostly common soil microbes that we've seen previously, but nobody had really done this work in cedar glades, so it's nice to know what some of that bacterial diversity is within the cedar glade. And the other nice thing about this project is, like I said, there's 60 different partner institutions that are all doing this kind of work. And so there's a lot of chances for students to connect across the country or internationally through Facebook, as well as our Small World Initiative website. So we have students coming and posting questions on Facebook, hey, this worked, hey, this didn't work, what can I do? And then getting feedback from a community of peers and scientists, just like you see in the actual academia. We also have a compendium of different universities that are all uploading the data that their classrooms are collecting. So we're getting this giant database of what's in the soil across the country and across the world, and what trends are we starting to see with regards to antibiotic production. So this crowdsourcing aspect is really appealing to students because it sort of brings in new technology in terms that the students are familiar with. And so I'm beginning my first forays into biology education, and so I wanted to see whether or not this model actually worked. I've been a big proponent of doing authentic research, but I didn't have the data to back it up. And so that's what I wanted to collect in this project as well. And so to do this, I surveyed 3 sections of introductory biology at MTSU. They had several similarities in common. So they were all honors sections. They capped at 20 students. 2 of them did the Small World Initiative model. One was non-majors, one was majors. These were the 2 classes that I was teaching. And then I also had a control group that was taught in the traditional manner with honors majors. So the way we've been teaching it at MTSU before I came along with this new model. And I wanted to generate different assessment data. So I want to know whether students are meeting their learning objectives, are they actually interested and excited about science, and can they start thinking like a scientist as well? So this is the first year preliminary data. We're hoping to run this experiment over multiple more years so we can start building on this. So these are just general trends I'm showing you at this point, but we're going to be building to this. And so I was doing different kinds of student assessment. So I have these surveys just to see how their opinions are changing about science. But I'm also trying to incorporate activities that are more like what researchers are going to be exposed to later on in their career. So beyond just writing the typical lab report, I also made students prepare a scientific poster, and we had a virtual poster session. So 2 examples of those are at the top and the right. So they were able to show to the rest of the MTSU community, this is what I did all semester. I invited faculty in that had no knowledge of the Small World project. They all got to ask questions to the student, and the student got to show off how much they'd learned. We also had students do research presentations, which is down in the bottom left. We have a group that just talked about one of our tester strains, but did it in the form of an episode of House. So you can see that grumpy student in the corner with a cane pretending to be House. And the kinds of products that they produced were actually really impressive. So this is a poster prepared by one of my Small World majors, and I told them that this is where they can really express their creativity. And so while there's a ton of information on here, it's a lot different from the typical 3-column kind of poster that you typically see. And so they really enjoyed this aspect because they got to be very creative. And so like I said, we're surveying these students across 3 sections as well. We're generating assessment data. When I'm collecting this data, I tried to keep as many things controlled as I possibly could. So all of 3 of these sections were honors, like I mentioned. All 3 of them have an integrated lab and lecture, which allows for a little bit more flexibility with how we're approaching the class. The major difference is that the non-Small World class that was our control was taught by a different instructor than I. Next year we'll be running another honors non-majors section being taught by a different instructor as well, so we can add to that data set that way. And I used a variety of different assessment instruments in order to determine whether or not this was working. So I did a short biological concept instrument survey. This was just, are they getting basic biology content knowledge? We did a class bio survey, and this one's great because it helps students perceive how science and STEM works. We had a project ownership survey, so are students do they feel like they're invested personally in the project that they're doing? And then also we have a sparse test. This is a survey instrument that we're still working on, but it looks to see whether or not students can design an experiment, interpret results, understand primary literature, things like that. And so here's just a survey or a sample of some of the results we've collected. These are all questions from the class bio instrument that we ran. And to orient you to these, we have a 5-point Likert scale. So scores of 1 means strongly disagree, scores of 5 mean strongly agree. And then you can see a couple of symbols on these graphs too. The squares represent classes that are majors. The circle represents the non-majors class. If they're in blue, that means that they had the small world model. If they're in red, it means they were the control group. And that little diamond that you can see also shows the aggregated pre-course ranking. So where did all of the students involved in this experiment see themselves before they did anything, and then at the end of the semester, how had those opinions shifted? So you can see that in some cases, our classes were similar pre and post. So curiosity about the living world led them to study biology. All of my different classes agreed somewhat with that statement. But we start to see major shifts in other fields too. So learning biology changes my ideas about how the natural world works. Everybody started moderately agreeing with that statement. My students either increased or stayed around that same average, whereas the control group seemed less interested in understanding how the natural world works by the end of the course. Additionally, the idea to learn biology, it's all about memorization of facts and definitions. At the beginning, the students didn't really think that was the case. My students tend to still not think that's the case. The control group is much more focused on memorization at this point, which is a little bit disingenuous in terms of trying to understand how scientific processes start to work. And again, these are preliminary data. This is with a cohort of three different classes. So these might shift a little bit as we build a larger data set. This is just the first trends I'm seeing. And so our first conclusion is that it seems like our small world participants are more interested in process and experience rather than just memorization, which is great. Our second set of data here, again from the class bio, is about civic engagement and personal responsibility. So whether or not they were asked whether the general public misunderstands biological concepts, middle of the road at the beginning. My students are shifting more towards agreement on that. The control group is less focused on that concept. This one's really interesting. So biology that is not directly relevant to human health is not worth my time. So the way I interpret this one is initially everybody said they're not particularly worried about human health. And what I think is being shown here is that a lot of my students are more focused on academic pursuits, graduate school, PhD programs, and research, as opposed to just everybody wants to be a pre-med and go into medicine and be a doctor. And so it's a big shift here that was really interesting. And then also trying to understand this contribution to society. So what I find is that my majors class is actually more interested in contributing to society relative to the non-majors or the control group here. So there's that giving back to society trend starting to emerge. So hopefully students are starting to see the civic engagement value of authentic research. And there are many other survey questions. This was my last one that I want to show you just because I like it. So would you take another biology class outside your major requirements just for fun? So would you take a biology class just as an elective? Middle of the road to begin with, and my majors by the end of this class, all but I think one student said strongly agree, I would definitely do this just for fun. I think that some of my non-majors were a little burnt out at the level of content, so they might not want to do it again. But the control group was also not that interested in adding in these extra courses. So hopefully we're getting the Neil deGrasse Tyson effect, where he can't hear you over how awesome science is. When it comes to assessing student outcomes, this data set is particularly small, so the only trend that I really care about at this point, the only statistically different trend, is noticing that our students are still meeting the learning objectives. So if you look all the way on the right, you have the dotted line representing where the pre-data was, and then the different dots representing where our different classes are after the post-survey. And so we find that regardless of the manner of instruction, be it traditional or small world, all students have a similar gain in terms of meeting those learning objectives. So they're still getting the content, they're still able to do scientific writing about the same, And then in experimental design, this is sort of an odd trend. It's a non-significant trend. My majors actually performed a little bit worse on the experimental design. Largely, I attribute this to low response numbers on this part of the survey. I think I had about an N of 4 for that class as opposed to an N of 7, and the other classes had between 10 and 11. And so that's a trend that I want to see if it keeps up over time, but I think that it's gonna shift as we get a larger data set. So we're not losing that content using this model, which is important. So overall, this project is allowing for project ownership, real experiments. Students are learning more about written and oral communication through posters and peer-reviewed lab reports. They're preparing themselves for future classes. My students that are leaving and going into microbiology classes are a lot more ready and prepared to actually be able to do things like the aseptic technique and maintaining cultures and things like that. And they're excited about it. Students came to lab every day because they wanted to see the next step in the process. And even if they weren't doing so great in the lecture portion, they still were really interested in what was going on for lab. And so you had that engagement, and you want this ability for students to want to stick around, which was great. And so future directions for this project, like I said, we've got about 60 different institutions. We're collecting a lot of pedagogical data across the university landscape. We have this giant aggregate data set for research purposes in terms of where are we finding putative antibiotic producers. I'm stationed within our honors program, and so with our honors students, they have to complete a thesis in order to graduate with honors. And we have freeze-back cultures of the bacteria that they've isolated. If they want to pursue the chemical aspects of this further, we have the samples available for them to do that. And then a new initiative that I'm getting involved with is our Small World partners are starting to look into whether or not we can use this system in a high school setting. So this coming year, I and one other partner are going to be teaching this in high school settings to see whether or not it works for them as well. And that is the end of this. Thank you very much.